Hello, my dear friends. It's Snacks Biology, and I am Hemant Naik, your biology teacher. Today, we are going to discuss about the transforming principle. ये आपका जेनेटिक्स का एक चैप्टर है, मोलिकुलर बेसिस ऑफ इनहेरिटेंस का, और 12th क्लास के लिए ये बहुत इंपॉर्टेंट क्वेश्चन है 5 मार्क्स के लिए, और 2 मार्क्स 3 मार्क्स में भी क्वेश्चन आ सकता है. Suppose if I able to clear your concept very simply, then don't forget to subscribe my channel. Okay, let's start. Frederick Griffith, one of the greatest scientists who given this principle. उसका मतलब क्या था उनका? ये मतलब था कि वो जेनेटिक मटेरियल को कैसे एस्टेब्लिश करे. तो बिफोर दैट व्हाट इज द कंफ्यूजन एक्चुअली? वी हैड इन द प्रोटीन्स, वी हैड RNA, we had, and DNA we had. Actually, what is the genetic material? At that time, it was totally confused. At that time, he taken this experiment by help of a bacteria. And what is that bacteria? Streptococcus pneumonia. Streptococcus means what? It is a spherical shaped bacteria is called the streptococcus. And because it causes the pneumonia in the organism, that's why pneumonia, Streptococcus pneumonia, the bacteria name. What is the strain? Strain means when we can divide one species of bacteria in different categories for the experiment point of view. They having the different biological activities and physiological activities. Like that, these two bacteria having the different biological activities they have. What is the biological activities? One is different from other. First, before knowing the principle, you have to understand about the R strain and S strain. R is for the rough and S is for the smooth. R strain means we also called R2, because why this number R2, 3 means they having the different cultures in the lab, and the particular species they are taken for the experiment. This one maybe it was R1, R2, R3, but R2 only he had taken for the experiment. That's why taken as R2. And it is non-capsulated. What does it mean? Non-capsulated means if you will see the bacteria, it having the cell wall. This membrane is called the cell wall. And this one is called the outer membrane, which is called the capsule. That means above the cell wall there is a membrane which is present in some bacteria, that is called the capsule. It is non-capsulated, means R strain doesn't have that capsule. And because it doesn't have the capsule, no polysaccharide coat is in. What is the polysaccharide coat? This capsule is made up of a substance that is called the polysaccharide. You know that polysaccharide what it is. And because it doesn't contain the polysaccharide, it is a non-virulent. What is the meaning by non-virulent? Means not causing the disease. What type of disease it will cause? It will cause the disease called pneumonia. Means it is called the cold and fever. After that, the organism will die. And what is about the S strain? S strain means it is the S3. We can call S3 strain. We can call this capsulated. That means what I explained to you. Above the cell wall there is a polysaccharide membrane which is called the capsule. That's why it is called the capsulated. And this capsule membrane, outer membrane made up of the polysaccharide coat. After that, this is the virulent. That means causing able.
Hello friends, now we will continue with the transforming principle. In the transforming one, we are now clear about that. There is a two strains are there. One is rough, which doesn't have the outer membrane, which is consist of the polysaccharides. That outer membrane here, what it is called? It is called the capsule. And here it is capsule is there, which is consist of the polysaccharides, and it is called the S3, and it is called the, what is the strain is called? R2, and it is called the S3. It is the virulent, that means it causes the pneumonia, and it is doesn't cause the pneumonia. This is the two different types of bacteria, but both are belongs to the streptococcus pneumonia. Now we will start the experiment. What he did, he take the S strain. S strain means what? That virulent. That means he is able to cause the pneumonia. Then what he injected into the mouse. He after taken the, because he having the two culture. If you clearly understand what is this, he having a big lab and it has the two cultures. In that S culture, one plate it is a S culture, and another plate it is R culture. That means rough culture, smooth culture. In smooth culture, he taken and injected into the mice. Then what happened? Mice die. Why the mice die? Because the mice is infected by the pneumonia, because the bacteria causes the pneumonia. In our case also, all the bacteria are not harmful or not causing the diseases. some are become harmful, but most of the bacteria which is present in our body are become very friendly with us. Without their bacteria, our digestive system will totally failed. That's why like that here also. R strain, which is a rough surface, that's why it is R strain. R2 also we can tell, and it doesn't have the capsule. And when it is injected into the mice, what happened? The mice live. Mice is not infected by the pneumonia. When the mice is not infected with the pneumonia, why the mouse will die? Mice will not die. Means mice is not affected by the pneumonia because this R strain is unable to cause the pneumonia to the mice. Now, third condition, what he did? S strain. Again, you take the S strain which is caused already pneumonia. But in this case, what he did? He heated that. When any microorganism we will get heated, then what will happen? It will die, because due to the heat treatment more than 100 degrees centigrade, no, it will microorganisms will die. When this heated, this assistant microorganism become died and injected into the mice, then mice is not affected by the pneumonia. That is mice live. That means he is not infected by the pneumonia, because when we heated the strain, the strain become healed by the heat treatment. Now what happened? The most important phase of the experiment, what he did, he combined the S strain, heat killed S strain, means the S strain which is already killed by the heat treatment, mixed with the R strain which is live. He is mixed in it. You will see here the S strain, what I marked S red color, they are S strain and they are R color, R which is a rough strain. S strain and R strain, they combined, R where the R strain is live and S strain is killed by the heat. When he mixed with each other and injected to the mice, then what happened? Then what happened? The mice died. What happened? You see, first when the S strain heated is unable to cause the disease. Only R strain unable to cause the disease. But when they mixed, they are able to die the mice. How? Because what happened here? The genetic material of S strain is a transport to the R strain. Although this S strain is killed, the remaining genetic material of the S strain, although it is a non-living, because he is killed, he is died, but its present of genetic material transport to the R strain. When it transport to the R strain, this R strain become able to cause the pneumonia, become virulent. That means able to cause the disease pneumonia, and the mice will die. Now you ask me one question, sir, how this S strain transport to the R strain, although it is heat killed? Because you see, HIV, which is caused by a virus, when the HIV will infect us, he is not going. He is only send its RNA or DNA, and that will enter in our body, and they will convert our DNA, behave as it's like that. Because our DNA will change into the behavior of the pathogen DNA. Like that same happened. Here, S strain is a pathogen, disease causing bacteria. R strain is not disease causing bacteria. When this S strain, the genetic material of S strain entered into the R strain, then R strains are become virulent and able to cause the pneumonia. And this pneumonia, by this pneumonia, the mice will die. What do you understand about this? So what is the conclusion of this experiment? This conclusion of experiment is that if a bacteria which is able to cause the disease and another is unable to cause the disease, the when we killed the microorganism or bacteria which is causing the disease, and we will mix with the R strain which is not causing the disease, but the genetic material which is present in the S strain is come to the R strain, and this R strain is able to behave as a S strain. That means R strain is able to cause the pneumonia to the mice, and mice died. And it is the first evidence that the genetic material of S strain moves to the R strain. That's why the R strain behave as a S strain. And it is the experiment which proves that the genetic material will flow from one organism to or moves from one organism to another. And I think you clear about the concept of the transforming principle. And it is the simple logic of that the genetic material which is moves from the one organism to other organism, like that the genetic material of a strain moves to the R strain and make the R strain causing the disease of pneumonia by which the mice died. Thank you for watching my video.